Hello and welcome to the second webinar in the 2017 Organic Seed Production webinar series presented by the Organic Seed Alliance and the Multinational Exchange for Sustainable Agriculture. This is your host, Alice Formiga of the eOrganic community at eXtension. You can find all eOrganic articles, videos, and recorded webinars on organic farming and research on our website at extension.org/organic_production and also on the eOrganic YouTube channel. The presentation will last about an hour and then we'll have about 30 minutes for questions. We'll be reading as many questions as we have time for after the presentation is over. So today we welcome back Jared Zystro, who will be telling us all about how to conduct seed trials and make selections on your organic farm or garden to improve the quality and performance of your crops. Jared is the Assistant Director of Research and Education at the Organic Seed Alliance, and he has worked in the organic seed industry for over 15 years. At the present time, he's also working toward his PhD at the University of Wisconsin, where he is studying plant breeding of organic sweet corn. So thank you for joining us again today, Jared, and I am going to hand over the screen control to you.
All right, thank you so much, Alice. I really appreciate you hosting this, and thank you to everyone who can join us today. So as Alice said, my focus for the next hour or so here is going to be on two parts of research, more or less, that you'll be doing that's fairly relevant to you, whether you're a farmer or just growing crops for market, or if you are a seed grower. These tools that I'm going to be presenting should improve the varieties that you have, help you find better varieties, and help you have a better understanding of how they might perform and might better perform in your farm. So this is broken into these two parts. The first part talking about variety trials, and the second part talking really just about the basics of how to do selection on your farm. So this first piece here is how to conduct a meaningful trial. And what I mean by that is that whenever you go out of your way to do a trial or do selection on your farm, really it is important to think hard about how to set up that trial in a way that at the end of the season, the investment that you've put into planting it and taking care of it and collecting information on the trial results in your ability to feel confident that you are able to decide which of these crops performs the best or has the attributes that you were looking for. And so I'm going to walk you through some of the steps to ensure or increase the odds that your trial will be successful. And what I would say is that when we talk about trials, there are many different reasons why you might want to conduct a trial. And this is really one of the reasons that I lump these two things together when I talk about variety trials and selection and plant improvement. Really, conducting good trials is a big part of doing good selection and variety improvement, and understanding the principles of what makes a good trial also will give you the principle you need to be able to do good selection and variety improvement. So there could be various reasons that you might want to conduct trials outside of, regardless of whether you are growing seeds or trying to do plant breeding. Trials can be useful for your farm just to be able to determine what varieties are out there that can perform well on your farm. Just because a variety, as most of you know who have experience in farming and gardening, just because a variety is advertised by the seed companies as being having certain attributes or just being all around stellar, that the many seed companies are, although they conduct their own trials, they may not be testing the varieties in an environment that's anything close to your environment. And so the varieties that perform well in the seed company trials may not be the same ones that perform well on your farm. And the best way to know which varieties are going to perform best for you is to try them. So another reason is to be able to screen through and look for varieties that might have interesting, novel, unique, or valuable quality traits. Looking for the sweetest carrots, the thickest walled peppers, the least spicy radish, or the most spicy radish. Whatever might be an interesting trait. The opportunity to rigorously compare the varieties side by side will help you feel more confident that you found the best of the available materials out there to meet those needs and display those traits. Another reason that you might conduct trials is if you are concerned about specific challenges on your farm. So this could be diseases, pests, or climatic challenges like heat stress or drought stress. Trials are an opportunity for you to be able to challenge a set of varieties with the disease or the pests or the heat or the drought and see which ones perform best under those circumstances. So I'm going to start by talking about just the first steps in planning your trial. And the first step I put here is prioritize your crops. And what I mean by this is that you will not most likely be able to grow trials of all the crops that you grow, either for seed or for produce. Be able to grow trials of all of those crops each year. And so thinking about which crops are the most important for you to conduct trials of, either because it's the most important crop on your farm or because it is a crop that has a particular challenge that you're really struggling with and you need to find a variety to overcome that challenge. These could be reasons why you might decide to prioritize a particular crop. What I would say really kind of across the board, whether we're talking about trials or we're talking about selection, is that you want to make sure that you only bite off as much as you can chew. You don't want to attempt to conduct 10 trials or 20 trials on your farm, and when push comes to shove, not actually have the time and resources available to successfully execute them in the middle of the season. And so being careful, especially as you're starting, about prioritizing and limiting the number of trials and focusing them on your most important crops will help increase the likelihood that at the end of the season, the investment that you've put into conducting this trial will pay off in some useful results. Second step here is to identify your goals. What do you want out of this trial? What are you trying to find? What specific attributes are you trying to find? What differences are you interested in? This will help inform what varieties you include in the trial and how you grow the trial and what data you collect in the trial. So understanding those goals is really important. And then with that information of understanding the crop that you're going to be working with and what the goals are for the trial, you can then go through and think about specifically when, what, and how are you going to evaluate the trials. And we're going to talk a little bit more about evaluation later in this lecture. And the next step, this is kind of winter work or early spring work, is as you're planning your trial, thinking about what are you going to include in this trial? What varieties are you going to be including? So this is really a critical part of conducting the trial, because on one hand, you want to make sure and include as much material as you can that you think could be promising. You don't want to, if it's not in your trial, then you won't have a chance to look at it and see if it really is the best. On the other hand, you don't want to include so many varieties in your trial that, again, you don't have the resources to properly evaluate them. So prioritizing what goes in your trial is a critical decision that you need to make. Some of the ways that you can do that are going through and looking to see, talking to your neighbors, flipping through the catalog, seeing what are the most kind of going to the grocery store, talking to the wholesalers. What are some of the most important commercial varieties in your region? What are people growing a lot of that because they perform well in your region and in your system if you're an organic farmer, and make sure that you include those in those. Those are important standards. Likewise, when we're talking about standards, going kind of outside of your region, what are some of the sort of most important national standards, the ones that are the best sellers that the seed companies provide? And if you are a seed grower and you're looking for varieties that you can potentially be saving seed of or producing for seed commercially, you would definitely want to be looking for what are the best open pollinated varieties. What are the best varieties that will be, if you save seed from them, will be true to type. That being said, I think in order to give you a realistic benchmark, if the most popular and varieties and the varieties widely considered to be the best are F1 hybrids, you want to include at least one or two of those in your trial so that you can have a realistic comparison to see, okay, are there any open pollinated varieties that are actually equivalent or surpass the performance of the best F1s. If you're working on a breeding project, you want to make sure and include your breeding material in there, both in terms of material that you're actively working on, and then potential parents that you think could be interesting to include in future breeding projects. And finally, if you are conducting a trial that involves some sort of test against a stress or a challenge, so whether we're talking about a disease or pest or drought or heat, you want to have some way to kind of bookend your trial. And what I mean by that is you want to try and find things ideally that are susceptible to that stress. Something that is well known to be, say you're trying to do powdery mildew squash, something that everybody says, oh, that thing succumbs to powdery mildew, if there's even a little bit of it around. And likewise, you want to include something that everyone knows is resistant. And this gives you a chance to make sure that disease or pest is present in your trial, and you get a sense of the severity of it, able to look at those susceptible and resistant varieties within the trial. So once you have your goals and you have your varieties picked out, it's time to think about how you're going to actually lay the trial out. And this is, again, something where you're going to be balancing your resources. So one of the things that science, in scientific research is common and is really applicable across the board, I think, for on-farm research as well, is the idea of randomization and replication. And really what this is about is it's about trying to minimize the risk that when you look at a variety, if you only plant it in one place in the field and it looks spectacular, it may be looking spectacular because that variety is genuinely superior to everything else in the field, or it may be looking so good because it's closer to your overhead irrigation, or it is where when you made a turn with the manure spreader, went over a bump and some extra manure dumped out in that patch of the field. There could be various reasons, various environmental reasons why a particular part of the field may be providing a little bit of extra help or a little bit of extra hindrance to your crops. And if you only grow something in a single part of the field, then you don't have a good way to tell if it's a variety is genuinely better or worse because of its genetics, or if it's because of its environment, because of its location in the field. So the idea of replication is that you would plant a variety in more than one place in your field. So when we talk about this, the lingo is reps. So you'd say, how many reps are in your trial? You know, a single rep trial means it's not replicated. Each thing is just planted in one place. You know, a 2-rep trial means that each variety would be planted in 2 different places in your field. And a 3-rep trial would mean that each variety is planted at 3 different places in your field. This on the slide, this says 3 reps is standard. That's certainly what you see often in kind of default in the scientific community for agricultural trials. That being said, there's a lot of reasons why you would use more or less. I would say that when organic seed alliance, when we're talking to farmers, I really encourage, you know, the idea of replication with the goal of you know, doing it again that's realistic, that is accomplishable with, you know, that it's better to do something than nothing, and that 2 reps, the jump that you can make from instituting replication, from going to just having no replication, from just having your variety planted in one part of your field to planting each variety in 2 places in your field, to having 2 replications, that jump is really important. And so I would say, you know, this says 3 reps is standard. Really, you know, if you can do 2 reps, it's really useful. And the reason that it's really useful is because you may not necessarily be able to have, you know, we'll get to this later when we talk about data analysis at the end of the season. You know, you may not be able to have what's known as statistically significant differences seen in your trial, but just having a second replication will be an opportunity to have a sense of how much variation is in your field. If you don't have a replication, then, like I said, you know, if something looks really good, then you don't know whether it really is really good or if it's just because it's part of the, you know, in a good part of the field. When you have 2 replications, all of a sudden you see, oh, well, it looks like this variety looks good here, but it doesn't look very good over there. And all of a sudden you get a sense of how much, what we refer to as error, how much error exists in this trial, how much environmental variation influences how these varieties look. And that will give you a certain amount of humility in terms of how strongly you believe the results, how strongly you think this variety is genuinely better than that. If you have at least 2 replications, you have a sense of, okay, everything, you know, these varieties really do look consistently, you know, this variety really looks consistently better than these other varieties, you know. And you know, I see that, you know, the varieties, each variety in my trial, I've got 10 varieties, and each time when I look at it, it looks the same, you know, in the 2 places that I planted it. So I feel pretty good about my trial. And if they don't look the same, you know, if each variety doesn't look very close in the 2 places it's planted in the field, then, you know, all of a sudden it gives you, you know, some measure of caution and doubt about interpreting the results of your trial. So randomization is the idea that in addition to doing this replication where you plant it, you know, in multiple places, you don't want to be planting those 2 things in the same, you know, just because you plant a variety, say, you know, you've got your honey nut squash, you know, and you plant it in 2 parts of the field, you don't want to plant both of those in the same part of the field. You don't want them both to be on, you know, say the outside row of your trial or right in the middle of your trial. You want to kind of random and mix them up so that way you have a better chance that the varieties are being planted in, you know, if there are differences within your field, that's being represented by the varieties. So this is talking about one of the ways that we can organize this is by using what we call blocks. And what this means is that if you look on this, this is again with where you would have 3 replications, where you would have where you, if you had 6 varieties in your trial, you would plant them. You plant the 6 in, you know, this yellow block. You know, 1, 2, 3, 4, 5, 6. You can see the 6 varieties are planted there, and then again, you have this red block, you know, which is further down the field, and you randomize it so that they're in a different order in this red block. And then in this blue block here, they're again in a different order. And this is pretty standard trial practices. The idea of this way is that if, say, moving from left to right in this field here on the left, if, say, it gets, the field gets more fertile, or the soil texture changes, that the varieties, that you have all 5 of the varieties kind of clustered close to each other, within each one of these blocks, and that they're all kind of sampling across the field. Depending on kind of how your field is, if you have a sense that there is a gradient in your field, in other words, if there is a known way in which the field gets, drier or more fertile or more shade from one direction of the field to the other, what you want to do is you want to put your blocks in a way that say all of the blue block would maybe be in the shadiest part of your field, and all of the red block is sort of in the middle shadiest, and all of the yellow block is in the sunniest part. The idea that the varieties within each block, as much as possible, you want to ensure that they are each experiencing as similar of an environment to each other as they can. In addition, if you have the space, it's really, really useful to include what we call a border row. This means rather than planting your trial right on the edge of the field, you plant it surrounded by that crop. So again, if we're talking about squash, you would have an outside bed of squash that wasn't part of the trial, and have that on each side, and likewise, include a planting in front of the trial and behind the trial. And the reasons for this, the most important reason really is that often the edges of the field are the most likely to experience different than the rest of the field. Those are the places where the irrigation doesn't quite reach, or the compaction from the farm roads that exist in your farm, kind of extend out to those outside rows, or because they're outside, they're a little more battered by the wind, or the first places that the pests and disease hit. So if you can kind of buffer and protect your trial by planting it in the middle of a patch of that same crop, it ensures that your trial is a little more likely to do well. So here's kind of a picture, looking at your field. If you're trying to set up your blocks, and you can see, in this field that there is some variation in terms of the soil conditions, you would try to arrange your blocks in a way that, if you look at this, maybe, this is sort of a dry patch at the top, and then there's sort of like a wet, low spot in the middle, and then dry again at the bottom. Try and line up your blocks in a way that captured that. Okay, so another important piece of this is within your trial, as we've talked about again, in order to separate the effects of environment and feel more confident that the differences you're seeing in your trial really are because the varieties are genuinely genetically different, you want to try and make sure that your whole trial gets as consistent of field conditions as possible. So part of this is just thinking about a part of your farm where the soils are, as uniform as possible. Although you can attempt to try and capture things with these blocks, it's better to really just aim for a spot that from the past is really, a uniformly, decent patch of ground that doesn't have, an old gravelly riverbed running through the middle of it, doesn't have, some big oak trees, up against the side of one of it. Trying to find something that's, as uniform as possible for soil type, for irrigation. And then when you're managing the trial, you want to try and manage it as uniformly as possible. So what I mean by that is that, if you're irrigating, if you're cultivating, if you're fertilizing, all of these things, you would like to try and do them in the same way to the whole trial at the same time. What you don't want to do is say, get started weeding, and get through, a few of your, what we call plots. A plot is one planting of a given variety within your trial. You don't want to get through and weed a couple of plots and then, get busy with your farming and come back to the trial, 2 weeks later, and those spots were the only spots that got weeded, and then try and go back and with the trial, because all of a sudden now the performance of those plots, that got weeded early when nothing else did, they may perform differently just because they got a different management. Same thing goes for all these other conditions. This all being said, one thing that you want to be careful of is that, although you want, to do your best to give, to find a uniform patch of ground and treat it uniformly, and, not, have those border rows so it's not on the edge of the field. You don't want to baby this trial in a way that you wouldn't grow your crop normally. You want to be still growing this as if it was a production crop. You want to be, cultivating it as often as you would, applying the same amount of fertilizer as you would, for a production crop. You don't want to give this, management, way above and beyond what you would already be doing for your crop. Because a lot of times, ultimately, if your goal is to find a variety that does well in your conditions, then you need to make sure that the trial is being grown in those kinds of conditions. And in particular, one of the things that we've seen in that, scientific literature has seen over time, over the history of agriculture experimentation, is that oftentimes when you grow crops in the most optimal of conditions, when they have, excess, fertility and, ample water, and, are, cultivated, meticulously all the time, and they're, grown just so that they're never stressed, that you may see less differences in those crops than you would if you put them through a little bit of, pressure, a little bit of stress, again, to the point that you would normally be growing your crop. So, in particular, if your crop is if you are looking to, if one of the goals of your trial is to be managing or finding varieties that are able to resist specific stresses, then think about, how you're going to, have those stresses exist in your trial. So if you're looking for something that's more drought resistant, you may want to reduce your watering. If you're looking for something that's disease susceptible, you may, or disease resistant, you may want to, plant, that border row of your trial with a variety that's, a susceptible variety to kind of encourage and increase the amount of inoculum that's, present throughout your trial. So those are things to keep in mind. You're not trying to grow, the most beautiful crop, and not stress your plants as, and again, I know as good growers, most of you are doing your best to give your plants optimal growing conditions. But what I'm saying is just don't go above and beyond what you normally would do, because then you'll have a harder time knowing how these varieties would actually perform in your farm. So once you've found your site, you've picked out your seeds, you've ordered your seeds, and it's planting time, how do you actually lay out and plant the trial? So first thing is, you're planting your varieties in these little blocks. So these can either be, or these plots, and these can either be, planted, one plot, represents a certain fraction of a bed, 10 feet of a bed, or it could be a certain number of rows wide by a certain number of feet long. It might be maybe 3 rows wide by 10 feet long. You want to be consistent in terms of the size of each of those plots. And so, you can see, tape measures, or your friend here, you can kind of measure it, mark it out, which, where each of those plots is going to go. And then you can either, ahead of time, make your map for how your trial is going to be. You can make it on paper or spreadsheet and determine where each variety is going to go. Or you can do it, in the field at the time. You could just say label these stakes, and kind of, for each of your blocks, throw the stakes up in the air for each of the blocks to randomize them, and then, put them, put them in place in the block, and then plant according to what the stake says. That being said, whether you prepare your map ahead of time or you, develop your map after, it's really important to have a map. Stakes alone for keeping track of your trial, or flags alone for keeping track of your trial, is very risky. You want to have a backup somewhere of what is where. Ideally, something on paper and digitally, because stakes break, Sharpies wear off, things happen, and then all of a sudden you've got plots that you don't know what is where. So the stakes and flags are useful because it helps you to kind of quickly see what's where as you're walking through the field, but you want to have a backup. Depending on what crop you're growing, you may want to have breaks between the varieties. You may want to have a gap where nothing's planted, so that way, as either something like a vining crop when we're talking about squash, so that way the plots don't get all tangled up with each other. You can separate what plot is where, or in the case of a really big tall crop that really fills up all the available canopy, that way you still have a pathway to get through to look at your trial in the mid-season. The flip side of having breaks is that those are places that can get weedy, so thinking about how to manage those what we call alleys, the breaks between each of the plots, is something to keep in mind. That's going to be a little bit different than if you're just growing a solid block of a single variety. So there's various ways again to mark the trials. You can use stakes, you can use flags, you can use these helpful little hangers here, and it's often convenient to have a to both number and name your varieties, so that you have a quick way to be able to write something on the stakes and refer to it. Additionally, by numbering things in the field, it allows you to collect data and look at the trial blind, as we say, which means that you can collect information about the varieties without bringing any of your biases into the trial ahead of time. If you already have an idea of whether it's because it's your favorite variety or your own breeding material, if you've already got some that you've got some biases around things that you want to see do better, then by keeping it blind, by just having numbers and not knowing, not memorizing what numbers refer to what names, when you collect data and when you walk the trial, you can have a more honest recording of actually how things are performing. So moving on to how you evaluate the trial, again, this comes down to thinking about what data you want to collect, when you want to collect it, and how you're going to collect it. And again, back to my original point, you want to ensure that you don't overburden yourself with so much data collection or such intensive data collection that you don't actually get the information that's most important to you. So thinking about when to evaluate a trial, oftentimes there's more than one time that you're going to be evaluating a trial, because there's different things that you can see that there may be the best time to be able to discern a particular trait at different points in the season. If you're looking for seedling vigor, you're going to want to be looking at the seedlings to differentiate that. By the time the plants grow to full size, differences in early season vigor, may have disappeared. Likewise, if you're looking, you may be caring about something, on the far side of the trial, like storage, durability of your squash, in which case you're going to be, that's not something that you can evaluate, when you initially harvest it. You're going to need to be planning on storing things to be able to, evaluate that over time to see how well they store. So it's really important, as you're making your initial plans for the trial, to understand, when the best time is that you want to be able to collect particular data about particular traits, and make sure you've got that kind of marked on your calendar, and that you're that you're keeping track of it, as the crops grow, because each season's different and the growth rates are different. You don't want to miss those windows, because you only have one chance each year to evaluate a crop during a particular window. So what are some traits that you might want to evaluate? There's, this list can be as long as your imagination can provide. Things like height and stature, earliness, yield, color, flavor, texture, storage, uniformity. All these are things that are pretty typically evaluated in a trial. And so there's kind of two different ways that, well, I guess 3 different ways that you would actually do an evaluation of a trial. That we use all 3 of these pretty regularly in our trials at Organic Seed Alliance. So one of them is the idea of scoring your trial or rating your trial. And this would mean basically applying a subjective rating. So, this could be on a 1 to 5 scale or a 1 to 9 scale, where for a particular trait, say, plant, well, that's a good example. Maybe root shape for carrots. We see some carrots in the field here. For root shape, where the ugliest looking roots get a 1 and the nicest looking roots get a 9. And this is a subjective thing, and so there's a couple ways to try and increase the odds that you're going to be able to make good scores. The first is by setting the trial, which means that before you go through and rate all the plots, just walk the trial and look for what you think is the worst looking, or the plot that has the poorest rating for a given trait, and that's going to be your 1 for the day. And then look for your plot that has the best, is the best for that trait, and that's going to be your 9 for the day. And then look for something that's in the middle, that seems to be kind of smack dab average, and that's going to be your 5. Once you have kind of that gestalt for what a 1 and a 9 is and what a 5 is, you can then go back to the beginning of your trial and start giving things individual ratings. Another thing that you can do is ahead of time write out kind of how you would describe your scale. So you could decide, okay, this is what a 1 means, and write out a description of what that you think that trait, what you would give a 1 for that trait, what reasons you'd give a 1, likewise a 9 and a 5, and all the numbers in between. You can also take advantage of having a photo of what you gave a 9, what you gave a 5 for, that you can then refer back to later. The second way that you can collect information about particular traits is measurement. And so measurements could be things like using a ruler. It can be things like using a ruler to measure height, or in this case looking at ear length and width on sweet corn. You could be measuring weight of yield, and the pro of measurement is that it's objective, and it takes some of the mental burden off of trying to really decide, is this a 5 or is this a 3, because your scale or your ruler just tells you what the number is. The minus of it is that often measuring can take more time than scoring, and if that's the case, if it's something that you can score relatively easily, and feel relatively confident about the robustness of your scoring, if it can save you a lot of time to do that instead of measuring it, that just again encourages you and increases the likelihood that you're going to collect the data on the trial. So think hard about what things you need to measure. If you set yourself up for having to do a lot of in-depth, complicated, labor-intensive measurements on all your plots throughout your trial, ultimately, if that means you don't have the time to do that in the middle of the season, then you might want to step back and think about, is there a way for you to score for that trait instead? Finally, the other important part of collecting data is notes and photos. It's just as important as these numbers. You really want to make sure that you give yourself room and space to collect notes for things that don't fit cleanly into your existing scoring system or your existing data collection system, where you can really note, why you gave something a particular rating, or something that stands out that thought to collect data on, but you want to make sure that you record for your future self when you're reviewing this in the fall or winter. Likewise, taking photos of the plots of important traits really can give you a lot of useful material to refer back to later. So keeping a record, so these, you can be collecting this data on data sheets. You can be writing it, in something like this Write in the Rain notebook. You want to make sure, again, get this data whenever you're collecting it, get it out of the field, ideally get it, off of paper and scanned into a computer, just take a photo of it with your cell phone if you got good resolution. You don't want to go through all this effort and then, lose your notes, get them rained on, have something happen to them. So again, back up this information as fast as you can. So at this point, now you're getting to the end of the season. How do you make sense of what you've seen in your trials? Kind of the simplest way is just looking at the means of the plots. So if you took, your variety and how well it performed for each trait, and you averaged that across all the different replications that particular variety was grown in, that would give you the mean or the average, and that's a useful piece of information. It's also really useful to get a sense of the error in that number. In other words, how much variation was there, and I'll show you an example on the next slide. But getting the error, that may require statistical software, though Excel can provide you a pretty good sense of something like standard deviation. That will give you a sense of, how confident should you be in a particular number? How much, did it vary from plot to plot? If you want to get more involved in kind of a sense of, were these varieties actually statistically significantly different from each other, then I would say contact me, and we're actually trying to set up a system right now that will allow you to kind of easily enter your data on the web and see some of that information. So here's an example kind of, for a particular trait. So this would be carrot top height. You collected these scores, and, looking at it, it's really useful to have all the replications out there, and look at them together. Seeing the averages is a quick way to kind of glance through and say, okay, which one performed the best? So it looks like here in this case, Scarlet Nance had the best tops. But then second, place was Bolero here. However, if you look, across this, if you see, in the case of Scarlet Nance, it was pretty consistently good in all the reps. An 8 in the first rep, 7 in the second rep, 7 in the third rep. Well, there's Bolero. The first rep got a 7, second rep got a 9, but then the third rep, something happened. And this is why, looking at error, looking at the results from the individual reps and taking good notes is really important, because hopefully, you may have something in your notes that gives you a sense of why that plot of Bolero was so short. And it may be, a environmental reason that leads you to, want to not include that result from your third rep in the average, or there may be something that really, where Bolero's response to particular conditions was odd and is important to take into consideration when you're deciding which varieties you want to carry forward. So then there's kind of, you can visualize now across all the different varieties and all the different traits to get a sense kind of overall of, if these were the traits that you cared about and that you measured, across all those traits, which ones seem to be holding up the best for the traits that you care about here. So this is a kale trial, we're looking at, vigor, uniformity, flavor, disease, pest susceptibility, and you can kind of get a sense, okay, overall, which ones, performed well across the board. Some of them, really stood out for particular traits, but then really fell down flat for other traits. And, this information can kind of, at a glance, help you as you decide which of these do you want to continue working with, which of these do you want to bring on to your farm, which of these do you want to think about saving seed or doing breeding project with. Last thing I want to end with trials here is that trials are ongoing. So as all of you know, the new varieties are coming out each year, so it's always good to be able to kind of take a look and refresh your knowledge of a crop by conducting new trials. In addition, a single year's results are not necessarily the be all and end all. Different years have different environmental conditions. The variety that performs the best in a hot year might not perform the best in next year when it gets cold. So by conducting trials for more than 1 year, and it both gives you an opportunity to look at new material, and it also increases your confidence in the results that you saw. So, you want to think about, as you move forward with trials, if something's looking really lousy, you don't need to keep trying it out forever. You can start, dropping those from your trials. And I would finally end by saying that the Organic Seed Alliance, we have a lot of useful resources. We have an on-farm variety trial guide that I would encourage you all to download. It's available for free from our website, seedalliance.org. We also have trial worksheets that are really helpful that I use personally to collect data, that are really easy to fill in the blanks and use them in your data collection efforts. So the next part that I want to talk about is maintaining and improving varieties through selection. So this is talking about, this is really kind of carries forward from trials. If you're finding that you have a need for an improved variety that doesn't exist on the marketplace, or if you are stewarding seeds that you want to ensure they continue to be good and that variety is what you want it to be and even gets better over time, then understanding the basics of selection is really important. So selection, when we talk about selection, this is ultimately in the context of seed production, what we're talking about is we're talking about selecting the plants that you will allow to make pollen and or to make seed, deciding, okay, these are the ones that are going to be contributing their genetics to the next generation, and these are the plants that are not going to be contributing their genetics to the next generation. Why bother to do this? There's various reasons, but really a big part of it that I always say when we're talking about seed saving is that when you are seed saving, you are, like it or not, you are a plant breeder. When you're talking about an open pollinated variety that is able to, cross-pollinate, it's endlessly generating new variation, new genetic variation. And within that variation, there's going to be some plants that are better, that are more like the ones that you want to have in that variety, and there's going to be ones that are worse. And if you aren't actively selecting and doing a good job of maintaining that variety through selection, then over time those varieties will tend to deteriorate, because natural selection in many cases is at odds with what you care about. For example, in sweet corn, which is a crop I work on a lot, sweetness is determined by the degree, the efficiency of how well the kernels of sweet corn can convert sugar into starch. The kernels that are the plants that are less efficient at converting sugar in their kernels into starch are the sweeter ones. It takes them longer. They don't convert as much sugar into starch, and so they tend to be sweeter when you eat them. What that means is that it leaves the kernels with less starch in them, and so when it comes time to plant your sweet corn, kernels tend to be slightly poorer to germinate, whereas the ones that are starchier, that were not as sweet, that those are the ones that tend to germinate better. So if you just plant out your sweet corn seed every year, the seeds that are most likely to come up and the plants that are most likely to grow vigorous tend to be the plants that are less sweet. The ones that are more sweet tend to have less starch in their kernels, and the seeds don't germinate as well. So if you want to maintain a sweet corn, you've got to be actively selecting for sweet plants. And the same thing goes for lots of different traits. So, why would you select? Some plants might be inferior. They could be smaller. They could be disease prone. They could be poor yielding. Or the plants just might not match a particular varietal type that you're trying to ensure that this variety matches what you think this variety is supposed to to look like and taste like and grow the way it's supposed to grow. It's supposed to be a bush zucchini, not a vining zucchini like this picture. So, by selecting, you can maintain it to type. So part of managing selection is on the preventative side, just making sure that you're not introducing unwanted mixtures of unwanted pollen or unwanted seed into your variety. So you can avoid unwanted pollen by making sure that your crops are properly isolated from your neighbors, from any volunteer plants from a different variety of that same species that could cross with it. And if there's any weedy crops or any weedy plants that are the same species, so like Queen Anne's lace in the case of carrot, is the same species as carrots and can cross pollinate. So avoiding and making sure that no pollen's coming from those, is one way to avoid unwanted genetics coming into your variety. Likewise, avoid unwanted genetics coming into your variety at the seed level by being very careful in managing your cleaning. If you're harvesting multiple lots, multiple varieties of the same species, make sure that you're very careful in maintaining your equipment as you clean and as you harvest. There's a few things I want you to take away from this talk today. And one of the key kind of next level concepts for seed growers who are looking to improve and maintain their varieties is the idea of stock seed. Stock seed is what really allows you to start being more aggressive in your selection, in managing your variety through selection. Stock seed is the idea of growing seed specifically to be your seed that you're going to be using in the future to grow your future seed crops. This is as opposed to production seed, which is the seed that you produce to sell to everyone else, that they're most likely just going to be turning around and planting to grow vegetables or grain or whatever that the crop the seed will produce. Once you have the idea of this lot of seed, this field, this part of this field is going to be for stock seed, then all of a sudden you have the ability to invest more time, effort, energy, thought into selection within a smaller field, because you don't need as much seed for your own use. So when we're talking about selection, when does it make sense to do selection? Again, your time is limited. How do you strategize around when to select? So part of it, again, going back to our trials talk, part of this is thinking about when can you best identify differences between your plants in terms of particular traits. Again, kind of thinking about what traits are you evaluating, what traits are you selecting for. If it's early vigor, then you want to be making selections. You want to be going out in your field and picking the plants that you want to be allowing to reproduce at the time that they're small, when you can see the differences in early vigor. If it's again in the framework of going all the way on the other end of storability, you want to make sure that you're evaluating and selecting and have enough plants and fruits left around that you can be doing selection in storage. Say your squash. One of the things that is important to keep in mind, with that kind of overall rule of trying to select when you best can distinguish differences between plants, but as much selection as you can do prior to pollination, the better off you are. So if you're able to, if you plant your crop a little bit on the thick side and you thin out all of the least vigorous plants, if you have a disease or insect pressure that shows up, early in the season, and you can eliminate any plants that are susceptible to that before they're able to begin pollination. Anything that you can do before they pollinate means that you have the opportunity to eliminate both that plant as a source of seed in terms of contributing genetics the next generation, but also you can eliminate that plant that's inferior before it can pollinate. So you also eliminate its pollen from the next generation's genetics. And that increases your efficiency of selection. So when we're talking about selection, I kind of broke it down pretty simply into these three broad categories. We talk about, and I'll talk about these more in a second here, but talk about negative mass selection, also known as roguing, talk about positive mass selection, then we'll talk about progeny or family selection. So when we talk about negative selection, what we're really talking about is this is really kind of a matter of degrees. The difference between negative selection and positive selection is really just a matter of degrees on a continuum and a matter of perspective. What this is usually what we're talking about is a relatively low percentage of your crop is going to be removed, is going to be excluded from contributing seed or pollen to the next generation. And this is something that can be done even when we, going back to those terms we just used, even on your production seed, even on your production crop, you want to be roguing or conducting negative selection there. You want to be walking your field looking for varieties that are, looking for plants that are off type or are inferior, or are showing, pest susceptibility or are smaller than the rest or whatever that is, and removing them from the field, hoeing them out before they can pollinate, hopefully, or, pulling them out later in the season, as appropriate. But, we're talking, somewhere in the range of maybe, 3, 5, maybe 10%, of your plant. So much that, you can do this in your production crop without hurting your yields that much. On the flip side, when we're talking about positive mass selection, this is typically where you are selecting relatively few plants to produce pollen or seed for the next generation. So you might be, out of a field of, 1,000 plants, maybe you're only selecting, 100 of those plants, 10%, or even, 50 of those plants, 5%. So you're selecting relatively few plants out of a large field to be the plants that you want to contribute to the next generation's genetics. And this again is where, thinking about, production seed versus stock seed is valuable, because you can, if something you are determining, that this is your stock seed production, you already have kind of, it's going to be a smaller field. You're not economically dependent on it yielding well for you. So you can afford to be pretty aggressive in your mass selection. You can also, one of the things that's pretty typical is you can basically select your stock seed out of your production field. So for example, if you had, a large field that was, you're producing seed for sale, you would go through and you would do your negative mass selection, your roguing, you'd be removing your worst things, the things that you don't want, you definitely don't want in your variety. But then you'd be going through that field and flagging your very best plants, the plants that perform the best, or in the case of a biennial crop like onions here, you'd be going through all of your bulbs and finding your very, very, very nicest looking bulbs. Say out of again, out of maybe 1,000 bulbs that you pulled out of the ground, 100 of those bulbs are no good for whatever reason. They're too small, they're too ugly, they're you're not going to replant those at all. And then 800 of those are pretty good. They're going to work. They're good enough. And those are going to be the ones that are going to be for your production seed. And then there's another 100 that are the cream of the crop, and those are the ones that are going to be your stock seed. And so you can whether it's in the field or in the cooler, in the case of biennial like this, out of the ground or in the ground, you can be selecting your stock seed out of your production field. So what are some tips for doing mass selection, this positive mass selection? Well, first thing, I guess, to understand it as we, to define our terms to move as we move into the other progeny or family selection. When we're talking about mass selection, what we're talking about is selecting based on an individual plant. We're looking at a plant in the field and we're saying, we're looking at a bulb on the table and we're saying, in and of itself, does this look good? Does this not look good? Does this have the traits that I want or not? Am I going to save seed from it or not? It is a fairly straightforward, simple selection process. It has the advantage, because it's straightforward and simple, that you can, potentially be growing a large population. Some of the tips for doing mass selection, as I talked about before, as much selection as you can do before things pollinate, the better, because then you're eliminating both the source of pollen and of seed of something you don't like. As large of a population as you can meaningfully, do a good job of managing and selecting from, the better. Is uniform of conditions, just like when we talk about the trials earlier, the more that the differences that you're seeing in the variety of the plants in your field are because the plants are genuinely different and not just because the field is really variable. So that the more uniform you can make the field, the more confident you can be that plants are actually looking better because of genetic differences. And last is a strategy which is selecting from quadrants of the field equally. And I'll show you a picture to describe that. So for example, in this picture, if say you do have kind of a gradient, say of fertility or soil, where maybe over in the lower left-hand corner it's more gravelly, and up in the upper right-hand corner it's a really nice silt loam. And rather than just picking the very, very best plants out of your whole field, in which case you kind of all be picking from this upper right-hand corner, if you mentally divide your field into pieces, in this case into 4 pieces, and you try and pick the very best plants from within each of those sections, you have a better chance of picking the plants that are genuinely genetically the best plants. So the next step in kind of complexity of selection, this is really something that you may not do all the time, but is a very, very powerful tool, and is something that will really allow you potentially to step up your game if you need to improve your variety or you're developing a new variety. And really what we're doing here is we're just trying to turn our selection into a trial. We're trying to, just like we talked about before, picking out your very favorite plants, saying, flagging them, saying these are the ones I want to save seed from. But the difference comes in when it comes time to harvest, rather than just harvesting all that seed into one big bag and replanting it the next year, you're going to be saving seed from each plant into separate individual bags or individual containers. So if you found 20 really nice plants, you're going to be saving 20 separate little bags of seed. And now you basically have 20 entries in a trial that you're going to plant next year. So the next year you're going to be planting each of those families, each of the, seed from each of those bags into separate little plots. Just like in the trial, like we talked about doing trials, you're going to be planting those in separate little plants next year. And then as you walk through your field, you'll have these individual plants. If you have the room, again, it would be ideal to actually replicate that. So plant, each of those 20 families, 20 seed from those 20 separate bags of seed that you saved from 20 plants the previous year. Plant those in more than one part of the field to replicate. And then what you're going to do is a 2-step process here. What you're going to do is you're going to try and find your very best families and select those. And then on a family level, on this plot level, whichever ones are the poorest, you're going to eliminate those. You're going to, ideally, try and do this before pollination. You be hoeing them all out. You're going to end up with a field that looks kind of funny because you've got, these just, a bunch of, missing rows within your field, or missing plots within your field where you've, hoed or rototilled or whatever the plants out. And then you'll have these occasional rows of the families that look good that you're selecting. Then within those families, within those plots that are, the best looking families, you're going to be selecting the best plants within those best families. So, in this case, we're selecting, these 3 plants are the nicest looking plants plants within this family that was the nicest looking family. What you don't want to do is you don't want to be tempted by, say in, this family in the middle, if all the plants look crappy in that family, except there's one that looks, that looks good, but, the other 5 plants do not look good, don't select seed from that, because you have an opportunity. These plants are all related to each other. They all, shared the same, maternal genes. They all came from the same, from the same other plant. And so you have an opportunity to basically see that plant's siblings, and that will give you a sense of some of the genetics that may or may not be currently appearing in that plant. So again, just because you see a nice looking plant, if the rest of the plants in that family look really bad, eliminate that whole family. Only save the best plants from within the best families. And at that point, this is a way to really kind of turbocharge and speed up your selection process, because you're able to turn this into a trial where you're able to grow something replicated. You're able to grow whole plots, so you get a better sense of how these things look, and you're able to see not just an individual plant, but also its relatives to get a better sense of its genetics. So that is, again, kind of just a super brief overview of selection. What I wanted to mention here before I open this up for questions is that as part of this Beginning Farmer Rancher project and the organic seed internship, one of the things that we wanted to offer is an opportunity for folks to get a more in-depth instruction on some basics of seed, but also on organic breeding. And we have a really great opportunity if you are anywhere near the Davis area in California, or within a drive. We're going to have an all-day organic breeding and seed intensive on August 10th in Davis. And this is a great opportunity. We're offering it for a very low price, basically just, I think it's $25, which is basically enough to cover lunch. You'll have lunch provided. And we're going to have some of the best organic breeders in the country coming out. We're going to have Dr. Bill Tracy, who's a sweet corn breeder, and Dr. Julie Dawson, who's worked extensively in wheat, but now does a lot of work in many vegetable crops. And I'll be there as well. And we'll be talking about, a lot more detail around selection and breeding and seed saving. And one of the great things that will be available as a resource there is that this is going to be in conjunction with what's known as the Student Organic Seed Symposium, which is a conference of organic breeding graduate students. And they all sent in a whole bunch of very fascinating breeding material from their organic breeding project. So we'll have all that available to us at the Davis Student Farm, so that we can walk through the fields. We'll be able to do hands-on demonstrations. We don't know at this point, what's going to be mature, right? We're hoping to be able to do some hands-on pollination demonstrations with a number of crops, but also just an opportunity to really hammer home the components of how to do selection and breeding with some real life examples of cutting-edge organic breeding research that's happening. So if you're interested, take a look at seedalliance.org/events. Look for this organic breeding and seed intensive and register. If you have any questions about it, feel free to send me an email at jared@seedalliance.org. And thank you all so much.
Thanks, Jared. That sounds fantastic. We're going to open this up for questions now. And if you have a general question that's not specifically about organic seeds, you're always welcome to use the e-extension Ask an Expert service. And this is for questions about organic farming specifically, and you can get an answer. So at this point, we don't have a huge number of questions, but we did get one that some people might be interested in here, about how the breeding method that you described is different from genetic engineering.
Sure. So that's a great question. What I'm talking about is when we talk about selection, this is something that humans have been doing with plants for a very, very long time. Basically, almost all of the cultivated crops that we eat are thanks to our ancestors selecting for traits that they cared about, selecting for larger seeds, or grass where the seed doesn't shatter and fall on the ground before humans are ready to harvest it, for squash where the fruits aren't as bitter and have thicker flesh. All of these things are in part thanks to the efforts of humans doing selection over a really long time. The same kind of process, is what I'm talking about. When I'm talking about about progeny and family selection, this is something that has been done as well for a long time, but is definitely has been less commonly used and is a very useful tool. But it's fundamentally something where we're working with the plants, finding the plants that do the best, and saving seed from those plants. The difference between what we're talking about and genetic engineering. Genetic engineering has a variety of definitions out there, but fundamentally what most people are referring to when they're talking about genetic engineering is the in vitro kind of meaning outside of a living organism, the removal of of DNA, of the genetic code of organisms, and the manipulation of that in a test tube, to be kind of oversimplifying it, but and then the reinsertion of that DNA back into a plant. So the idea that you're in some way manipulating the genetic information, that the DNA, and then inserting it back into the plant is certainly different than what I was talking about when I was talking about doing selection. All of the selection that's happening here is still, plants are still, reproducing, using their existing, flowers, and they're, producing seed, in the way that they normally do. The humanness is really coming in terms of us deciding, which seeds we're going to be saving seed from and which plants we're allowing to pollinate in our field.
Okay. In the case of breeding depression, what is the best method for selection?
So typically, I think what you're referring to is more commonly called inbreeding depression, and so this is inbreeding depression is something that occurs most commonly in crops that are cross-pollinating crops. These are crops where they have various mechanisms to encourage crossing between different plants in that same crop. So something like squash, for example, where you have very conspicuous big flowers that are encouraging bees to carry pollen from plant to plant, or corn, the wind is blowing the pollen around. These outcrossing crops, for kind of evolutionary reasons, rely on genetic diversity to maintain vigor. If just like humans, really, humans were an outcrossing species, and what happens is if there is not enough genetic diversity in the population, you start to see more and more negative, deleterious, genes that start to appear, and those can result in smaller plants, smaller fruits, less seed, etc. And so the biggest part of reducing your risk of inbreeding depression, of maintaining genetic diversity, of maintaining what we call hybrid vigor that exists when these outcrossing crops have enough genetic diversity, the biggest piece of this is population size. What I mean by this, and I would encourage you to take a look at Organic Seed Alliance's seed saving guide, where we talk about population size and give some good numbers for each crop, is that the population size is how many plants should you be saving seed of in order to maintain that genetic diversity that is necessary for that particular crop to not start to become inbred, to not show inbreeding depression. And so, as you're going through these breeding projects, it can be tricky because, normally when you're growing a seed crop, you can say, okay, well, I need to have, 50 plants to maintain my population size and maintain my vigor maintain that genetic diversity, okay, well, if I plant 60 plants and I, rogue out 10 of them, I still end up with 50. But if you're trying to do this kind of, aggressive selection, whether we're talking about that positive, we're talking about the family selection, you may need to be starting with much larger populations. You may need to start with 500 plants in order to cut it down to 50 at the end, and still maintain that vigor. That being said, one of the things that you can take advantage of is there's often a little bit of a window, I would say, where you have, you may be able to be more aggressive for a single year in terms of dropping that population size down below what's optimal for a year, to do some of this more intensive selection. But you definitely want to be aware of that and careful of it in your breeding. That is definitely a risk factor. By trying to very aggressively select, that you end up inbreeding your crops. Again, that matters more in outcrossing plants than in some things like, say, tomatoes or peas, that you could be selecting very few plants and not see inbreeding depression.
Yeah, it can be really a challenge when you only have a few seeds of a particular variety, because I remember I worked for a seed company once, and people were trying to increase the seeds of a very rare morning glory, but it had been pretty inbred, and so it was very unvigorous, and so it was really a challenge to multiply those seeds and have them be good enough quality to sell.
Yeah.
So we have a question about storage of seeds and how to manage moisture.
Okay, so briefly, again, I would take a look at our Organic Seed Alliance's free seed saving guide. You can get it from our website, seedalliance.org, under our publications page. But some real quick guidelines is that ideally you're managing. When you're managing for storage, the things that you want to manage for is you want to manage for moisture, you want to manage for temperature, you want to avoid your seeds coming in contact with water, you want to avoid pests being able to get to your seeds, and you want to avoid fluctuating temperatures. So when we're talking about moisture in particular, some strategies there. I mean, obviously, if you live in a place where the ambient humidity is really low, then you're in luck, and that's pretty easy. Then it's just a matter of making sure that you're keeping your seeds, from getting, rained on, that they're in a sheltered, location where, no moisture's, no actual, water's getting to them. But if you live in a place with high humidity, like I live, you usually you have kind of two options. One is that you create environmental conditions, and you basically make like a seed room. You create a space that is where you're controlling the environmental conditions, potentially with something like a dehumidifier, and in that case all the seed that you're storing in there exists in that environment. The other option that's I think a little easier, especially at small scale, is that you create that you're basically just manage the humidity within the individual containers that your seed is stored in. So if you've got something that has an airtight seal on it, like a mason jar, or like a tote that you taped up the edges of, what you can do is first make sure that the seeds that are going into your container are very dry and are fairly clean, because chaff, the non-seed material, tends to act as sort of a sponge and soak up water. But if you can get your seeds to be clean and dry going into the container, that's the first step. And then you can put something like desiccant in that container with you, with the seeds. So if you've got something like silica, I'm often looking at something on a 1-to-1 silica to seed ratio, or maybe 1-to-2 or something like that. You can just put that in the container with the seed. You don't want to mix it right in with the seed, but put it on the bottom of the container, and then have your seed within like in a breathable little pouch or something like that, also in that container with the desiccant. That will help manage the humidity inside of those individual containers.
Okay, and one more question about rodent protection when storing seeds, some different ways to do that.
Yeah, that's a tricky one. So I would say, I mean, again, kind of the easiest thing, so a lot of it depends on your quantities, right? So if you're talking about, saving seed on a, home scale, or saving seeds of something like, tomatoes or peppers, where, or even brassicas, where you may just have small quantities, then, looking at, metal, glass, things that are, naturally rodent proof, are going to help with that. When you're dealing with larger, larger quantities, again, I mean, it doesn't hurt to think, rodent proofing. I've seen farmers build kind of seed bins that they've constructed out of plywood, where they're basically these big sealed plywood boxes that they can screw in a plywood lid on top of this 4x4x4 container, and mice can't get into it. The other thing you can do is again sort of manage your seed, the seed space, where the rodents are. So trying to put your seed up on shelves, and try and create some kind of little barrier at the bottom of each shelf, some little poles that are hard for the rodents to climb up, and setting some traps within that seed room, are some other options. But it is a hard one. I would say, for myself personally, I find it best to sort of work on a container by container level to make sure that those are rodent proof. And you can actually, what you can do too is you can, again, if your seed quantities are small, you can put a bunch of containers that may or may not be rodent proof into a single larger container that is rodent proof.
Okay. All right, well, thank you. We are running out of time. I'd like to thank everyone who submitted questions. So thank you so much, Jared, again. And we hope everyone can join us for the other webinars in this series, which take place on the third Tuesday of each month through October. So thanks, everyone, for joining us.